Thank you very much for this kind introduction, and also I'd like to thank the organizers and specifically Dr. Ayman for the invitation, but also for putting such an amazing program. We've already seen fantastic talks on clinical aspects in rare diseases, then psychosocial, and then diagnostics, and so on and so forth. And I'm sure the rest of the program will be even more exciting. I will be talking today to you about delineating the functional implication of variants that are causing rare diseases with the potential for novel therapies. I will give just a short introduction, 1 or 2 minutes, about, for example, the demographic characteristics of the UAE population. As you all know, there are about 10 million people residing in the UAE, with the majority of them are expatriates, and therefore the population is multi-ethnic and diverse. The local population is characterized by the presence of isolated communities like the Bedouins, the Africans, Yemenis, Baluchis, and generally women tend to continue to reproduce well after the age of 40. And of course, as we had previously, the family structure is large and extended, and there is high level of consanguinity, more than 50% for some populations. This is the classification of the genetic disorders that are found in the United Arab Emirates in terms of their prevalence. And in fact, there are at least 400 different genetic conditions that have been described in the literature as indicated in the database curated by the Center for Arabic Genomic Studies, and the vast majority of them are autosomal recessive. The diseases in the UAE can be classified into common, including thalassemia, sickle cell disease, and there are conditions that are relatively common, i.e. they are more common in the UAE compared to other populations like Joubert syndrome, Meckel syndrome. And there are disorders that are clustered in a specific family or isolate or a tribe. For example, Stowe-Wiedemann syndrome is found in at least 50 families in the UAE, but they all belong to a tribe originating from Yemen. But obviously, the vast majority of those diseases are rare, or some of them are actually new, having been described previously. We at UAE have been using molecular, cellular, and genomic approaches, which led to the identification of at least 400 different disease-causing mutations underlying many conditions or tens of conditions, with various presentations, including intellectual disabilities, bone errors, metabolism, dysmorphology, and so on and so forth. We also identified several novel disease genes, and also we looked at the cellular mechanism underlying several conditions, and we elucidated them. Our approach, like many others, at least in the last 5-6 years, have been to ask the question whether there's a candidate or candidate genes that are associated with the phenotype in the child or the affected individual or the family, and then if there's a candidate gene, we'd go for Sanger sequencing. All this is now changing, as Dr. Abu Tayoun earlier indicated. We probably mostly now we go for next generation sequencing using whole exome sequencing or even whole genome sequencing. Obviously, we need to go further to elucidate the cellular mechanism for 2 purposes. One, to provide opportunities for therapies, but also to confirm pathogenicity. Finding a molecular change, i.e. a mutation, in a gene doesn't really mean that that change is pathogenic, and therefore functional studies are needed. What I will do, basically, as I said, we worked on tens or even hundreds of families. I will give just 2 examples or 2 cases that we worked on recently and came up with some conclusions. The first case is an Emirati family with 2 children affected with congenital heart defects or cardiac defects, cholestasis, jaundice, and failure to thrive. As you can see, the parents are consanguineous, and they have 2 children affected. One passed away, the other one is still alive, and I believe she received a heart transplant. As the title indicates, the children have a cardiac presentation. I'm not going to go over it. You do obviously limitation of time, but also they have liver presentation as well as dysmorphic feature or dysmorphology. So what we did, we performed molecular analysis, which which revealed a homozygous single nucleotide deletion at this particular position, at position 1058 in exon 6 of a gene that is encoding a protein called matrix metalloproteinase N or MMP15. This is the pipeline that we used to filter the data that was obtained by the next generation sequencing, and we confirmed the segregation of this mutation in the family. The child is homozygous for the loss of this nucleotide. The parents were heterozygous in comparison to a control. That mutation is found in exon 6 of the gene, which leads to a frame shift in the protein, leading to a premature stop codon or a truncation at this particular position, and that will lead to the loss of large part of the protein, including the cytoplasmic tail, the transmembrane domain, and several other extracellular domain. Therefore, this truncation mutation is expected to lead to total loss of function of this protein as a membrane or cell membrane matrix proteinase. MMP15 is a zinc binding into peptidase. Its major function is to degrade various components outside the cells or in the extracellular matrix, which is obviously important for organogenesis, including the formation of the heart and other organs. The matrix metalloproteinase have been implicated in a normal and pathological states, including in wound healing, tissue remodeling, and angiogenesis, and also in some tumors. Animal models of MMP15 indicated a major role of MMP15 in the endocardial cushion formation. And several other MMPs have been implicated in human genetic diseases. Because of the nature of the mutation, i.e. the formation of a stop codon or a premature stop codon, we hypothesize that this stop codon will lead to the instability or the degradation of the messenger RNA from that gene by a process called nonsense mediated decay. So we looked at the level of the messenger RNA in the patient, and indeed, or patient fibroblasts or cells, probably blood cells anyway. From the patient, you could see a drop or a significant drop in the level of messenger RNA in the affected individual. Also, the parents, the level of messenger RNA is slightly reduced. Meanwhile, at the same time, there had been another report of a mutation or a change in the MMP15 in a patient with similar phenotype to the patients that we are describing here, and that mutation is a single nucleotide substitution at this particular position, leading to a missense mutation, a glycine to arginine change. And we modeled that change using molecular modeling, and indeed we can show that there is a major structural change that leads to the loss of zinc binding, because this particular residue, this glycine, is involved in binding zinc, which is important for the catalytic activity of this enzyme, and therefore loss of this zinc binding will lead to loss of its function. The conclusion from this part is that the MMP15 is important in human development, particularly cardiac development, and also that its loss of function is the cause of this severe phenotype in the affected children. This work has been published just a month ago. The second example is, or the second case is basically an Emirati family with several children or 3 children affected by GM gangliosidosis type 1, which is an autosomal recessive storage disease. The index case is a male child, 7-year-old male, presented with developmental delay, muscle atrophy, regression with spasticity, and also scoliosis, recurrent respiratory infections, as well as uncontrolled generalized seizures. We performed whole genome sequencing using next generation sequencing, and that led to the identification of a homozygous single nucleotide substitution, or a single nucleotide substitution mutation at this particular position, which leads to a missense mutation at position 151, and aspartic acid to a tyrosine, as indicated here, and that's confirmed by Sanger sequencing. By aligning this, the sequence of this gene in humans and other species, we show that this particular residue or this aspartic acid is highly conserved from the fly or fruit fly Drosophila to humans, indicating that this residue is important either for the structure or the function of this protein. This mutation is located in the extracellular domain, as indicated here. These are other mutations that have been found in obviously other patients. We did modeling, molecular modeling, and indicated here, the blue indicates the native structure or three-dimensional structure of this protein, where the superimposed red structure is actually the one that is resulted from this particular mutation, the aspartic acid to a tyrosine. And you can see a major shift in the structure, and therefore expected to lead to loss of function of the protein. And indeed, we took fibroblasts from the patient, and we tested for this enzyme activity, the beta-galactosidase activity, or beta-gal. And here on this part, you can see the activity from cells derived from healthy individual. And this is the enzymatic activity of the sample derived from the patient. There's only about 0.5% of the original activity, indicating almost total loss of function. Here, I just want to indicate that this enzyme, this beta-galactosidase, we looked at its processing, trafficking, and targeting, and this protein is generally, like other proteins, is synthesized on ribosomes, in this case associated with the endoplasmic reticulum, and then passes through the critical pathway, i.e. through the Golgi, and then from the Golgi, it gets diverted to an organelle in the cell that is called lysosomes. Crucially, the movement of the protein from the endoplasmic reticulum to the lysosomes is associated with a proteolytic cleavage, which is essential for converting this protein from inactive or the precursor state to the active state, and therefore indicate that it is this cleavage or removal of this part is important for the activity, and also there is a drop in the molecular weight, which is important to understand the subsequent figures. So what we did here, we looked at the state of this enzyme in patients' fibroblasts, and we compared it with the fibroblasts from normal control. Here is the sample from the normal control, and when you probe it with the antibody against the beta-gal, you can see two bands, a high molecular weight band, which is the precursor, and this is the newly synthesized, the one that is in the endoplasmic reticulum, and this is the mature, the lower molecular weight band, which is the active band. Now, when you look at the sample from the patient, you can see only the precursor, the inactive, the mature band, and none or very little of the mature species, indicating and supporting that there is a total loss of function. This is a bit complicated figure, but what we did here, we looked at the subcellular localization of the protein, the wild type protein, from control cells as well as from cells that are derived from the patients or fibroblasts. And you need to take my word for it. Basically, what you see that this mutation leads to the loss of lysosomal localization of the beta-gal and its retention within the endoplasmic reticulum. We wanted to see if we can rescue this mislocalization issue or problem. And the first test that we did, we cultured the cell lines or the fibroblasts that we derived from the patients at reduced temperature. And that generally will allow some of the protein to fold, probably to attain its native structure. We also cultured some cells in the presence of a general molecule called glycerol that will also help proteins fold. Obviously, these cannot be used for therapeutic purposes, but they are for indicative purposes to indicate to us whether the protein or this misfold protein can be rescued. And indeed, you can see that the proportion of this signal or the mature band increases at 27 degrees or in the presence of glycerol. We also confirmed that by confocal microscopy, and again here you need to trust me that basically there is a shift, a little bit of shift in the subcellular localization of the protein in cells that are derived from the patients when we incubate them at 27 degrees and or in the presence of glycerol. That's really in terms of localization, but we wanted to look whether this partial rescue in the subcellular localization will lead to functional rescue at the enzymatic activity level. So we measure the enzymatic activity level in these cells that have been grown at reduced temperature or under glycerol conditions, and indeed there is a little bit of rescue. So the untreated or the cells that are being grown at 37 degrees untreated with glycerol, they would have a residual activity of about 0.5% of the original activity, whereas when you incubate at 27 or in the presence of glycerol, there's about 5-fold increase in the enzymatic activity, indicating that there's partial rescue at the cellular level, but also there is a bit of functional rescue. This led us to the question whether it would be possible to use what's called enzyme enhancement therapies using pharmaceutical chaperones to restore the activity rather than just growing the cells under in glycerol or under this reduced temperature, because obviously these cannot be used for therapeutic purposes, whereas pharmaceutical chaperones could. And the idea here is that you would add the pharmaceutical chaperone and would allow, probably would bind to the misfolded protein and will allow it to fold, at least partially, and then that will allow it to traffic normally to lysosome and restore some of the activity. We did a lot of studies on different molecules, and I just want to present a couple of those molecules, and these are of the basically galactose analogues, because obviously galactose is a substrate for this enzyme. And the two molecules that we tested are NN-DGI, which is N-nonyl deoxygalactoniggeromycin, or N-butyl deoxygalactoniggeromycin for short. This is in lysosomal storage disease. Similar molecules have been used, for example, migalastat have been used for treatment of Fabry disease, which is another lysosomal storage disease. So we wanted to test some of those molecules. And indeed, when we tested the first molecule, which is the NN-DJG, did not show any change in the residual activity, even after 3 days of 1 day or 2 or 3 days of treatment at different concentration. There was no change in the residual activity of the beta-gal in cells that are derived from the patient, i.e. the patient's fibroblasts. However, when we tested the other analog, the NB-DJJ, we found that it enhanced the activity by about 2 folds. Here is the untreated cells, the enzymatic activity. When you treat with increasing concentration of NB-DJJ, you have increased residual activity. That residual activity might not be enough therapeutic, but this is an indication that it might be possible to restore some of the activity using these pharmaceutical chaperones. Also, we looked at the processing of the mutant beta-gal, and we can see some accumulation of the mature form from the patient cells that are treated with this compound. This NB-DJJ-mediated enhancement was additive and persistent upon treatment with subsequent doses. For example, when we gave it for 6 days, basically you could see that this rescue of the functional activity or enzymatic activity is sustainable for several days, and also it benefits from dosing or giving the cells a new batch of the compound in day 2, day 3, day 6, and so on. And you can also see there is increased level of maturation and some increased level in the enzymatic activity. Again, this work has just been published, I think, last year. So the conclusions on this case too: loss of function in this family, which is responsible for infantile GM gangliosidosis, is due to trafficking defect in the protein, and its quantitative loss from lysosomes in the patient. There's some of the studies, or some our studies indicated that this mutant is amenable to rescue, which makes it suitable candidate for further studies for enzyme enhancement therapy. Then particularly probably around this compound, the NBDJJ, because it increased the activity by about 5-fold in some of the experiments. Obviously, further studies are required to not just confirm this, to expand this and make it therapeutic and potentially go for animal studies or potentially clinical studies. Just want to go back here, the general conclusions and remarks, and that ties well with what other speakers, including Professor Abatayoun earlier. Basically, employing combination of molecular cellular approaches was effective in elucidating the underlying mechanisms and the pathogenesis of several conditions. Obviously, these findings are very important for reducing or prevention approaches through carrier screening, through prenatal or pre-implantation genetic diagnosis, and also for implementing effective genetic counseling and also family planning. But also for the purpose of today's talk, understanding the cellular mechanism would give us new opportunities for developing novel therapies. As you all know, 95% of rare diseases are not treatable at the moment, and therefore we need to find new therapies. You might think, this is too much to go into the cellular and so on and so forth, but I'm afraid, to be able to develop novel and personalized therapies, we need to do these deep studies to look not just at the molecular level, at the genomic level, but we also need to dig into the actual mechanism, what goes on at the cellular level. Obviously, that might give us also clues for the prognosis in those patients. I just want to here highlight that we have recently established this UAU Genomics Laboratory with facilities in biochemical genetics, analysis, as well as molecular genomics and bioinformatics setup, including next generation sequencing, whole genome sequencing, whole exome sequencing, and then using bioinformatics tools to eventually reach some sort of a conclusion. We're doing all these studies on research basis. We are not a diagnostic labs like Dr. Abatayoun's lab, but we are obviously looking at those, especially those families that have no diagnosis when they go through the diagnostic labs. So we take them over and then hopefully try to provide diagnosis and evidence on pathogenicity, and in some cases go further and provide opportunities for therapy. Finally, I would like to thank my co-workers at the College of Medicinal Sciences, PhD student Hanadi Abdurrahman and Fida, who defended their PhD, and Hanadi will defend soon, and also my clinical collaborators, Professor Fatma, Professor Al-Hadh Al-Ghazali, and also my colleagues at the college, and also my collaborators who were involved in the data that I presented today, Dr. Muhammad Ghattas, Professor Osama Darbashi, Dr. Aisha, and Dr. Joseph Hertigan. And thank you for attending. These are some images of our work being displayed on major scientific journals, and thank you.